Good evening, everyone. I am Dr. Pooja, and today I will be discussing some of the very important questions on biochemistry. Let's start up with the discussion of the questions. Now, question number one is amino acid used in the synthesis of carnitine. First of all, you should understand what is the function of carnitine in the body, isn't it? So, जो carnitine है, this carnitine is actually required for the transport of the fatty acid from the cytoplasm to the mitochondria, because synthesis of fats takes place always in the cytoplasm, but oxidation या breakdown takes place in the mitochondria. So carnitine shuttle is required because whenever the body requires energy during starvation, during fasting, so this fatty acid will be transported with the help of carnitine. Okay, but यहाँ पे question क्या है? Question is the amino acid which is used in the synthesis. So the amino acid is lysine. This lysine is required for the synthesis of carnitine, and this lysine is an essential amino acid. Right? So here, if we see this particular question, two important points we can get. One is that lysine required for the synthesis of carnitine, and second is carnitine shuttle is required for the transport of the fatty acid from the cytoplasm to the mitochondria. So the answer to this question is A. Moving on to the next. Ninhydrin is a reagent used in the detection of. Now this is again a very important question. Always remember, एक ninhydrin होता है और एक biuret होता है. Ninhydrin and biuret reagents are the two very important reagents which are required to check the presence of proteins. It is a qualitative detection. And if we look at the principle of ninhydrin, it is simply decarboxylation of the amino acids. So the answer is amino acids, plain and simple. So the answer to this question is ninhydrin is required for the detection of amino acids, and that too qualitative. Moving on to the third question, Menadione is an analog of. Now always remember, this is a very important question from Viva point of view also. जो Menadione है, this is the only form of vitamin K, which is water soluble. This vitamin K has actually three forms. Out of the three forms, Menadione is considered to be water soluble. So it is an analog of vitamin K. So the answer to this question is B. Moving on to the fourth question, total number of dehydrogenases in TCA cycle. D माने removal किसका? Hydrogen. So if we look at only TCA cycle which starts from acetyl coenzyme A, like this, there are three dehydrogenases reactions. One is catalyzed by ICD, isocitrate dehydrogenase. Second is alpha ketoglutarate dehydrogenase. Right? Now, look at this question. If I talk about dehydrogenases, if I say NADH, so if I talk about NADH, it will be 3, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and the most important is malate dehydrogenase. And if we calculate about FADH2, it is SDH. Right? So it will be 3 plus 1. So total will be 4. So if the question is asked like this, total number of dehydrogenases as by NADH is 3. But if we calculate the total number, it is 3 NADH plus 1 FADH2. That is why if we calculate 3 NADH, so it will be 3 into 2.5 and 1 into 1.5. That is why it comes out to be total 10 ATP from one molecule of acetyl coenzyme A. So the answer to this question is C. Moving on to question number 5. Eye cell disease is associated with. Now this is again a very important question. This is inclusion cell disease, and it is associated with lysosomes. And the carbohydrate which is involved in this eye cell disease is mannose 6-phosphate. Because mannose 6-phosphate acts as an envelope. It's like an address on the envelope because proteins, once their function is over, they are sent for degradation to the lysosomes. So due to the absence of mannose 6-phosphate, eye cell or inclusion cell disease is caused. Which of the following is required as a coenzyme for the conversion of acetyl coenzyme A to malonyl CoA? Simple, acetyl coenzyme A has 2 carbon atoms. Malonyl CoA has 3 carbon atoms. That means we are increasing the number of carbon atoms. The process is carboxylation. And whenever we talk about carboxylation, the coenzyme required is always biotin, which is B7. Always remember, B7 is required always for carboxylation. So the answer to this question will be D. Then moving on to the next question. A non-competitive inhibitor does which of the following? देखो, non-competitive, no competition between the inhibitor and the substrate. So inhibitor can go and bind. This is a very interesting question. Inhibitor can go and bind with the enzyme, or it can bind with the enzyme substrate complex, whatever may be the case, because there is no competition. If there is no competition, obviously there will be a change in Vmax because the velocity is going to decrease. But if we talk about Km, there won't be any change in Km. So Km will be unchanged. Now you look at the options. Where is this option? Vmax decreases. Yes, Vmax is never increased. Decreases Km and decreases. So answer will be decreases Vmax. So the answer will be A. Right? Next question. Which of the following is a energy requiring step of glycolysis? Energy requiring mane ATP is being consumed. So always remember, we know glycolysis has 3 phases. Energy investment phase. So you just look for those reactions in which energy investment phase is there. That is simply glucokinase or hexokinase, where glucose gets converted to glucose 6-phosphate and ATP is consumed like this. So it will be glucokinase. If you look at other reactions, phosphoglycerate kinase, energy generation phase, 2 ATP are synthesized. If we talk about pyruvate kinase, 2 ATP are synthesized. Lactate dehydrogenase, NADH is hydrolyzed. So energy requiring step is glucokinase and the answer will be automatically A. Moving on to the next question. Diagnosis of folic acid deficiency. Now this is a very important. Folic acid is benign. If you look at the option, obviously Figlu estimation is one of the very important tests. It is histidine load test. Yes, with the help of peripheral blood smear also we can check. ICAR estimation, again this is one of the very important quantitative determination of folic acid deficiency. So the answer to this will be all of the above. So the answer will be D, all of the above. Then moving on to the last question, glucose transporters in myocytes stimulated by insulin is. Now see, all of us are familiar with this. There are different type of glucose transporters. Some are insulin dependent, some are insulin independent. If we talk about insulin dependent are insulin dependent glucose transporters are 4, 8 and 12. Out of this, check the option, it is 4. So, glucose transporters in the muscles stimulated by insulin is GLUT4, right? So, these were the simple 10 questions which I have discussed, which have been actually asked in PG exam also. So, this video will be beneficial for all of you. So, if you guys like my video, please like and subscribe to my channel. In the future, I will be coming up with different more questions. So, wish you guys all the best. Take care. Bye-bye.